life, so many different life forms, so many kind of different ways in which we can do biology, so many different forms, functions. There is amazing biochemical and biochemical diversity. There's amazing diversity in form and function. And so it's really hard to believe when you think about it that all of the modern biology was really done on a sample size that equals 1. And the reason for that is because obviously there is only one life form. There's only one biosphere. All of modern biological life came from this one and only last universal common ancestor. So all of biochemistry that we study is identical. So despite this incredible diversity that we can see on the surface, if you dig deep down into the kind of core of our modern biochemistry, there's only one central dogma, and there's only one way to do biology. And so that's where this sort of controversial and aims to be controversial statement came from, is that all of modern biology has been done on a sample size that equals 1, because we only have one kind of a modern cell that we know of. And so that's obviously the problem when you want to learn some general properties of life, because you look at this incredibly complex cell and you don't actually understand it. Despite all the work we've done and all the progress we've done in understanding natural biology and understanding how life works, we still don't really understand where every single molecule goes in a living cell. And that personally bugs me a lot because I'm trained as a chemist, so I would like to know where my molecules go. And you can't do that as a biologist. You cannot have a biological system right now where every single molecule is known and where position and function of every single molecule is understood. And the funny thing about it is that even the simplest possible life form, the JCVI syn cell that has a chemically synthesized genome, still has few dozen genes that are essential genes of unknown function. So even if we synthesize the entire genome chemically, we still don't know exactly where every gene goes. And we still don't know what's the function of every gene and every molecule in that simplest possible cell. So as biologists, we're doing a pretty bad job trying to truly understand the chassis that we're working with. So this is the answer of people that came into that synthetic cell engineering field before me, is if you can't understand what you're working with, try to make it simpler. And that's the whole rationale of our field, is we're making synthetic cells that are simpler. It's a fully understood, controllable biological chassis that lets us study every element of a living cell in this much, much simpler and better understood, better controllable environment. And some of the things that you can study and understand in synthetic cells are things that you could never control and understand and manipulate to that extent in living cells. And so in a way, you can think of a synthetic cell as this sort of a jigsaw puzzle. You put together elements, biochemical elements of all different known domains of life, and what comes out of it is an entity that is very modular, very controllable, looks like a cell, smells like a cell, quacks like a cell, but is not living, at least not yet. And because we're putting it together in the lab, we can control every single element of that cell. So we can control exactly where every gene goes, where every protein, every membrane molecule. And so it's essentially a kind of lineage agnostic platform on which we can study natural biological processes, but we can also assemble new biological processes. And so notice that I still haven't really told you what is a synthetic cell. And the reason for that is because there's not really a consensus of this one particular platform is a synthetic cell. What we understand as a synthetic cell in my lab is a liposomal bioreactor that's capable of expressing proteins. And that's a very obviously expensive definition. We make lipid membranes that encapsulate a synthetic cell cytoplasm that expresses proteins from synthetic DNA circuits. And that's our chassis. What we do with it is, I'm going to show you within the next minute, what kind of functions we do with it is what really makes the system interesting, makes the system help us develop kind of new functionalities. And so the simplest possible definition of a synthetic cell that I think most people in the field would agree on is a synthetic cell is something that has not been alive before you put your hands on it. So there is not unbroken line of living organisms from the last universal common ancestor to your specific synthetic cell. Obviously, we're cheating because we're using molecules and proteins that came from living organisms, but they are dead by the time I get to them. And so we're building this organism, this artificial organism, from non-living components. And if I were forced to make a one-sentence definition, I would say it's a bioreactor that has some emergent properties. So it's more than a sum of its parts, but most of all, it's understandable and it's controllable. That controllability is the key. We want to know exactly what we're doing in this system. And some of the things that we can control are things that you could not otherwise control in living organisms. One of the kind of most common things that we play with that are impossible to play with in life systems is size of the cell, permeability of the cellular membrane, the identity of the cell-free protein expression system, so the identity of the translation system itself, and also the really small details, small essential details of the metabolism. And so, for example, you can engineer ribosomes, or you can engineer tRNA for the specific purposes. So again, this is what a synthetic cell is in my lab. And to give you a little bit of a molecular look on what we understand as a synthetic cell, the membrane, the membrane to me is a kind of a less of an important part. I was trained as a membrane biophysicist, so I should be more excited. But right now, the membrane for me is just a compartment. It's a lipid compartment made most of the time of phospholipids and cholesterols. So it's a membrane that mimics a natural cell membrane pretty well. And sometimes we use simpler, not modified fatty acids when we do more of a probiotic origin of life research. And then in that membrane, we often embed membrane channels for communication with the environment, for sense and response of environmental signals. You don't have to have a membrane. You can work with droplet microfluidic systems and create those millions of nearly uniform synthetic cells that can contain all of your synthetic cell cytoplasm in an aqueous phase, but do not contain a membrane. So depending on applications, depending on what you want to do next with your synthetic cells, you can, if you do it in a droplet microfluidics without a membrane, you have relatively easier experimental setup, but you don't get the benefit of being able to use all those transmembrane sense and response circuits. Now, what goes inside is the stuff that's really fascinating to me. And that is the synthetic cell cytoplasm, or otherwise known as cell-free protein expression system. So what is a cell-free protein expression system? It's not really cell-free. It's a protein expression system that came from living organism. In this case, at least what we're showing on this slide, it's a bacteria. So we take bacteria and we rip the guts out of it. We purify the ribosomes, we purify the tRNA, all the acyl-transferases. We put artificial DNA in, and what comes out is a protein product. So it's basically a way to make proteins in the absence of a living cell. And that is the central dogma that runs our synthetic cell systems. You might have heard of this technology if you have anything to do with metabolic engineering. This seems to be the kind of one of the fastest growing metabolic engineering tools that people use to assemble new pathways and troubleshoot pathways. And I'll show you some examples of use of that in a moment. You can have cell-free protein expression systems out of pretty much anything that you can culture. And so in my lab, most of the time we use bacterial cell-free systems just because they're the easiest to use. You can make cell-free systems out of more and more complex organisms. Obviously, as you do it out of eukaryotes or even mammals, the complexity of the proteins you can express increases. You get proper glycosylation patterns, you get proper other post-translational modifications, but the yields are going down significantly. And so for most of the experiments that I'm going to show today, we stick to the bacterial systems. So what my lab is doing is we're chemists that pretend to be biologists. We use the chemical reaction resolution to look at biological processes. So we're basically building those synthetic cells and we're assembling them to have a certain function and then do experiments with it that resemble experiments you could do with a live cell. And the idea of it is that we can learn how life is built, how life works, how life is engineered by putting it together one kind of subsystem at a time. And the reason we're doing it is, well, not just because it's cool, but also because it has a lot of actual practical applications. So these are some of the areas that my lab is interested in. For one, the most fascinating for me is this feedback loop between natural biology, tool development, and synthetic cell engineering. You need to know how synthetic cells work in order to understand natural biological processes that you put in them. But you don't know which biological process you want to use unless you actually understand the biology itself. So it's this feedback looping. The more we learn reconstituting processes, the more we can learn about the actual natural living cells. And then there's tool development. Some of the biggest achievements of the field of synthetic cell engineering so far also resulted in a really popular general synthetic biology tools. One of them is, I'm sure all of you who've ever done any cloning are familiar with the Gibson ligation protocols. And Dan Gibson developed that protocol in the process of working on the JCVI syn cell, the first synthetic mycoplasma cell. And so the tools that we use to reconstitute natural cells in the lab are also then later becoming a general use synbio tools. And that's kind of fascinating to me to see that feedback between things we do to troubleshoot and then things that become actual useful tools. And then there is a lot of practical applications that not just pay the bills, but are also exciting to work on. And some of the ones that I'm going to show you today is developing solutions for programmable, controllable biocomputing, doing some origin of life studies, space biology, which is my kind of private hobby, doing some metabolic engineering, which is my money-making hobby, and then the diagnostics and therapy applications, which is one of those new fields where the synthetic biology in general, and specifically synthetic cell engineering, is just really breaking into. Okay, so I gave you sort of an introduction to those liposome encapsulated synthetic cells that my lab is using. Now I would like you to forget about it for about 5 minutes. And I would like to show you an example of a project that I'm really excited about, but it has nothing to do with the actual synthetic cell. So remember the cell-free system that I was talking about? That's all we need for this particular project. So this is my first venture into the astropharmacy and space engineering. So the problem is that once you send people into space for a long periods of time, for example, to get to the moon or get to the Mars and back, they will get sick, obviously. They will get sick in a ways that we cannot anticipate in advance. And since there's no CVS on Mars, they won't be able to go and pick up a prescription. So we have to have a way to make drugs, to make whatever pharmaceuticals and whatever nutrients the astronauts will need on a short notice in a small scale and using a programmable biomanufacturing platform. So live cells are really bad for programmable biomanufacturing platform because live cells have some idea about what they want to do in life. And most of the time it is not making a drug for you. So to develop a strain that synthesizes what you need takes weeks to months. There's a whole industry around it, and for a good reason, because it's damn hard. So what we propose to do is use a lyophilized cell-free system. Why lyophilized? Because you can store it dry, you can store it at room temperature, and also you don't pay for the water on the launch. The atmos cost is a significant driver of all of the research in this field. And so it's been shown about 4 or 5 years ago by Jim Collins's lab that you can lyophilize the whole cell-free system on a filter paper. And they've shown that you can assemble cool circuits on this single piece of filter paper from a lyophilized cell-free system. So what we asked is it possible to lyophilize different elements of a cell-free system on separate pieces of a filter paper? And instead of having everything pre-programmed and then storing it on this one piece of paper, can we assemble a whole genetic circuit on different pieces of filter paper? And what you see here is a completely useless proof of principle circuit that we're using to troubleshoot that system. Obviously down the line, I would love to do metabolic engineering with it, but right now we're happy if we make a GFP. And so because I'm showing it to you, you can obviously figure out that yes, we did make a GFP. And so we went through all this trouble of lyophilizing a cell-free system on separate pieces of paper. And eventually at the end of it, we combined the papers rehydrate, and ended up with a GFP. It sounds simple, but from the kind of troubleshooting point of view, the difficult part of it was that how do you lyophilize something on a paper that remains intact during the lyophilization process, and then once you rehydrate it, all your layers have to match. So there was a little bit of a testing of every possible paper support in my lab for a while, just to find the right support. This is obviously, honestly, one of my funniest and favorite parts of this project. If you were to name a piece of a paper support that remains intact when you wet it once, but disintegrates eventually after being wet for a while, if you think about it, you will arrive at a solution and then you will discard it because you say, no way, I'm using that for science. But end up, this actually works the best. Toilet paper, tissue paper is the best possible support for this kind of work, because good quality fluffy toilet paper stays intact when we do a first round of lyophilization of everything. But then when we combine our sandwiches, we wet the layers, and that support disintegrates and combines all our enzymes and produces a product. So for a while, we had a testing of all different possible brands of toilet paper. Autoclaving a roll of toilet paper turns out quite of a fun adventure, because all of those experiments have to be RNase free. So we have to find a way to sterilize the toilet paper. Thanks to amazing grad student Adam, who did most of the groundwork on this project. And so now we have a working system where we can build those sandwiches of different circuits, different genetic circuits. I'm really looking forward to publishing it, because in the material and method section, I will be able to say toilet paper source Costco. So I'm really looking forward to getting more kind of useful circuits to work in the system. And this is an example of the useful circuit that we're working on right now. This is a vinylacetic acid circuit. If you've done any metabolic engineering, you know that this is the favorite pet circuit of everyone, because it's a circuit that you can create relatively easily, you can control it easily, and you can analyze it by HPLC. So this is where this work is right now. We're building more and more complex circuits that eventually, hopefully, will create something that makes sense, will create a useful small molecule. And so next thing is this well-known design, test, build, layer cycle. This is a kind of classical bioengineering cycle that people go through to design a new metabolic engineering pathway. And this part, so build part, make the plasmids, build it, put it in a bacteria, take the protein out of the bacteria, and then test your circuit, and then go back and redesign it. This build part is something that can be significantly simplified by using cell-free systems. So we're obviously not the first people to think about it. You can simplify the build part even further by skipping the cloning. In cell-free systems, you can express proteins from linear DNA fragments, so you don't have to clone at all. You can express your proteins, and the concentration of the protein depends directly on the concentration of the DNA fragments you put in there. So you don't have to play with strengths of different promoters and so on. So what we ask is, can you skip the build part as well? So basically, can you clone a whole plasmid in a cell-free reaction and express it in the same reaction? And so this is our experimental system. We're starting with a plasmid that has an artificial stop codon, and that gene on that plasmid is a good old GFP. It's actually an eGFP variant. A neat thing about GFP is that just by switching 3 amino acids that are in the, really 2 amino acids once you're cloning it, that are in the chromophore center, you can change the color of your GFP without touching the rest of the protein. And so what we did is we designed this plasmid that has a artificial stop codon where the chromophore should be, so there's no fluorescence unless we fix it. And then we fix it using a series of reactions, introducing a region that is inserting 2 amino acids into that chromophore, making a different protein every time. And this sounds like obviously a pretty lame cloning scheme until I tell you that this was all done in a single reaction pot. So we drop that plasmid into the vial or into the well of a 96 well plate, and then we sequentially add different enzymes, and we keep incubating it without any cloning. So everything happens in this one pot, and at the end, you have a protein expression in that same well. So all you have to do is keep adding proteins. There's no purification step at any time. So we're basically starting with one plasmid, and we can build an entire library of different, in this case, fluorescent proteins for proof of principle. And what we can do is we can clone different kinds of enzymes by mutating that one active center in the same reaction that we're expressing it. And so this whole cycle from cloning to expression takes about 12 hours, and it requires no purification. So you can imagine how that is going to be a really good tool for doing high throughput, fast protein evolution. And we've also done it now on antibiotic resistance, so on enzymes that hydrolyze different kinds of antibiotics. And we've done it on luciferase because we love model systems. So we develop different kinds of different brightnesses of different types of luciferases by modifying a luciferase active site. So this, the reason I'm showing this is because right now we're looking for collaborators for this project. We're looking for people that have a useful enzyme that can be modified by doing up to 5-6 point mutations. And what would you like to learn from that? We're tool developers. We don't know anything about the real science, the real world. So we need people that know biology and can help us with kind of figuring out what should we do with it. Okay, back to synthetic cells now. I gave you kind of an outline of those two useful projects. Now forget about useful projects. Let's focus on cool projects. So what I really like is when you study biology, and most of the time what you study, when you study what cells and populations do is they mate and they create new lineages. And so we decided to engineer that system into a synthetic cell. So what we did is we're starting with one population of synthetic cells, and we coax it to sequentially mate with another population, and then with another, and then with another, and then with another, basically creating lineages. So we're calling them pluripotent synthetic cells. They're pluripotent because they can differentiate into any kind of a lineage you want, as long as the lineage is obviously possible by the genome encapsulated by those synthetic cells. And as we like useless model systems, the first few model systems we created were for reporter proteins, but then we also demonstrated that it can work for metabolic engineering circuits. So the way we're doing it is we have this system where synthetic cells sequentially fuse with each other, creating bigger and bigger circuit inside it. And the way they fuse is using DNA sequences on their surface. So this system models snare proteins, which are the synaptic proteins that fuse your synaptic vesicles with the membrane of the synapse. So it's essentially a molecular zipper. You have those pieces of DNA that can be programmed to hybridize and induce membrane fusion. This is one example. The genes on this slide make no sense. This is a completely made up model system that we did to demonstrate that you can have 9 different populations of synthetic cells fused together, and only if all 9 of them are present, you'll end up with the final product of, in this case, firefly luciferase. This is proof of principle. This is the first time we've shown that we can do it. Once we have a toy, we're going to play with it. So just kind of a brief outline that we managed to modify the system in many needlessly complicated ways. We build a lot of different variants of it to show basically what are the limits of this combinatorial assembly. One of the things that biology likes to do is nonlinear pathways. So what we did next is we demonstrated that a single parental population of synthetic cells can differentiate into 2 different lineages, here marked by 2 different kinds of luciferase reporter proteins, depending on the events that happen in its non-living life. So basically what populations that parental population gets to mate with. And this obviously doesn't sound really impressive when you're used to biology doing it all the time, but please remember these are all artificial cells. Every single cell here was built by hand, and we know all the concentrations of all the molecules inside it. So this is a perfectly controllable system. Another thing we did is we demonstrated a speciation of those ancestral pluripotent populations that can be controlled by the identity of the populations that they mate with. So we basically have this system where we engineer a genome, a regulatory genome of that ancestral population. And during a series of those mating events, we create a final populations. The one thing that I really like about this system is that we simulated the outcomes of different fusion events, and surprisingly, the outcomes actually match what we simulated. That doesn't often happen in biology, I was told. You do simulations and then you do experiments and you have to reconcile because they don't match. We did simulations, we did experiments, and they match. That obviously made me super happy. Another thing we did is a useful, quote unquote, useful metabolic engineering system. So this is our old friend bioacetic acid system. We put it into our synthetic cell population. The nice thing about bioacetic acid system is that the identity of the final product depends on the order in which the enzymes were mixed. So here what you see is a complete bio pathway where the bioacetic acid is the main product and a CPA is a side product of that pathway. So that's what happens when you complete the pathway doing fusion of 5 different populations of synthetic cells. What you can also do is change the identity and distribution of the products of that pathway by fusing different kinds of synthetic cell populations in different order. So basically a take-home message here is that you can build in a controllable way specific metabolic engineering pathways. What we're doing right now is we're trying to do something even more useful. We're engineering the system to contain other metabolic engineering pathways that actually provide products or provide a pathway discovery tools for finding new pathways that we didn't know about. Okay, so here's another very quick, the reason I put that on this slide is not to show you any cool results because we're early enough that there's not that many cool results yet, but to show you the capacities of the synthetic cell system. So what you can do in a synthetic cell system is a lot of things, but you cannot kill it because it's already dead. And the nice thing about starting out dead is that you can't really get any more deader. While in live cells, if you kill a cell, you have no cell. So that makes the system perfect to use for studying things that you cannot really study in a live, breathing, living cell. And one of those things is a ribosome. If you know the basic biology, you know that the ribosome is really a terrible catalyst. It's really an entropy trap. It's not an honest catalyst. And so we cannot evolve a ribosome to that extent in live cells, because if you dysregulate any essential part of the ribosome, you end up with a dead cell. So you don't really have anything to study. If you do it in a synthetic cell, though, you have a wide margin of slightly semi-dead, but still making some protein you can detect areas. And that enables us actually evolving on the critical parts of the ribosome that have not been touched since the last universal common ancestor. And 2 of the parts that I would like to point your attention to are the ribosomal hinges and the peptidyltransferase center. Both are RNA, so we're not evolving ribosomal proteins here at this point. We're evolving ribosomal RNA. And so you can evolve ribosomal RNA in this selection scheme where we use a small protein, very small reporter protein to detect activity of a really broken ribosomes. And what we're doing right now is we're evolving the ribosomal hinges from a library of ribosomal RNA, and we're evolving a new functionality of the PTC, the peptidyltransferase center. This shows you that you can do things with synthetic cells that you couldn't really do with live cells, because you would, instead of getting an evolved cell, you would end up with a dead cell. So this is kind of a more versatile bioengineering system. Now, another thing you can do with synthetic cells, and that's again, more showcasing the capacity of the platform rather than showing a specific experiment, is that you can make things inside a synthetic cell, and then you can export something to the outside. So just first, very, very brief proof of principle. You can make things in synthetic cells, and that thing in this case is a fluorescent reporter protein, and then tag it with a cell-penetrating peptide tag. These are the good old cell-penetrating peptide tags that you know from 10 years ago when people thought that's going to be the next drug delivery thing. It was not the next drug delivery thing, but the tools remained, and they can carry a payload across a membrane. So what we demonstrated is what you see here is a size exclusion purification traces that show that with a cell-penetrating peptide tag, a membrane of the synthetic cell, so that's the top 2 graphs, remains intact, but the protein leaks outside. The protein is in this case GFP, so it's fluorescent, we can trace that. Without the cell-penetrating peptide tag, again, membrane's intact, that's our control, all the protein sits inside. Okay, Okay, so now we can export proteins out of a synthetic cell. What do we do with it? Well, one of the things we do with it is we can carry a payload. So we engineered a protein that carries a RNA tag. So it's a RNA binding protein with a cell penetrating peptide tag, and the protein is made inside a synthetic cell. It drags this RNA payload outside, and we can measure that by measuring the presence of that fluorescent RNA payload. It's a fluorescent RNA aptamer that we can measure the presence of it outside. So we can basically make something inside a synthetic cell and drag it outside. Now we can also make something inside a synthetic cell and drag it outside and then get it into another population of a synthetic cell. So this is two populations of synthetic cells at this point exchanging a protein. In this case, the protein is a T7 RNA polymerase. So in the absence of the T7 RNA polymerase, the so-called population GFP has a gene for a GFP, but cannot make it because it's under T7 promoter, and that population doesn't have a T7 RNA polymerase. The other population expresses the T7 RNA polymerase, but has no reporter. So only if that T7 RNA polymerase is expressed from the population T7 and then exits the population T7 and enters the population GFP, you see an increase in fluorescence, which indicates in synthesis of the GFP. So this is an example of two different populations of synthetic cells exchanging active proteins. Now, another thing that we can do if we have synthetic cells that can communicate with each other is looking at possibility of building organelles. So some of the things we're doing is looking at membraneless organelles, but we're also looking at using natural cells as organelles for synthetic cells. And so in this case, I want to show you how you can co-culture a cyanobacteria with a synthetic cell. So basically what we have here is a synthetic cell that has a cyanobacteria encapsulated inside it. And so that synthetic cell does its own protein expression thing, and it has a cyanobacteria inside it. Why cyanobacteria, you might ask? Well, because cyanobacteria were engineered to secrete lipids into the environment. This is one of the strains of cyanobacteria that was engineered back when people thought that this is going to solve our energy problems. We're going to make biofuels in cyanobacteria. We're not making biofuels in cyanobacteria, but we have those strains that can secrete lipids. And what you see on a video is a growing synthetic cell. So this is a dead synthetic cell. It's growing as a result of the cyanobacteria secreting lipids. And those lipids are then going into the membrane of a synthetic cell, and that synthetic cell grows. It grows into those threads because of the osmotic imbalance between the inside and the outside environment. And so we see that there is a living organelle inside a non-living synthetic cell. So another example where synthetic cells pretend to behave like a living population is we can mimic antibiotic resistance. So we have two synthetic cell populations. They both have a GFP reporter gene. They both have an alpha-chemolysin membrane protein, and one of them has a kanamycin resistance. So alpha-chemolysin is a membrane channel necessary to bring in the kanamycin into the cells. GFP is obviously a reporter. We add kanamycin to this environment, and what happens is that in both cases, the membrane remains intact. So the synthetic cells are physically still there. What you can see on this microscope picture is the bottom two panels. Red is a membrane. The membrane of the synthetic cells was dyed red using a membrane dye. So we have that red labeled membrane, but only in presence of kanamycin resistance, we still have the metabolism of the synthetic cell going. So we can see that the GFP is being expressed in that synthetic cell that has kanamycin resistance. Again, sounds trivial when you think about doing it with bacteria, but this is not a living system. This is a completely engineered synthetic system that shows traces of antibiotic resistance. So another cool thing that you can do is you can try to make a cell-free system from the simplest possible living cell, and that's the Mycoplasma Syn 3.0 cell. The reason I'm showing just one slide on this project is we're just finishing paper 1 on this project. That basically the kind of take-home message is that the simpler the system, the harder it is to create a cell-free system from it and reconstitute the entire working system back up. But we're on our way there, and the goal here is to take a genome of the Mycoplasma synthetic cell, put it into a non-living artificial cell that has the mycoplasma cell-free protein expression system, and see if we can jumpstart the actual mycoplasma. So now that's the end of real biology. I would like to, at the end, show you just a few kind of a speculative fun projects that we're working on. So one of the things that you can do with a synthetic cell is you can write down the composition of that cell and send it to someone else. And this is one of the kind of coolest applications if you want to ruggedize it. If you want to use synthetic cell to create remote pharmaceutical solutions, remote biomanufacturing solutions. And so if you can write down an information about engineering a specific synthetic cell and then send it to a remote location, as long as you agreed before on a set of parts you're going to use, you can recreate any particular kind of a synthetic cell. So that remote location can be obviously on another continent, but it can also be somewhere where you cannot deliver. And so that's the basis of all the astropharmacy solutions. That's the basis of our work on trying to demonstrate that synthetic cells can actually work in space and can actually be used as useful biomanufacturing tools. And speaking of space, here's another kind of a crazy project we're working on, and that basically is trying to put a quantitative threshold to aliveness. So when you think about that, if I ask you what's the definition of life, most of you will try to give me a definition, and everybody will be wrong because we do not have a working functional definition of life right now. This becomes really useful when you want to go out and look for life. And so what we're doing is we're building synthetic cells of increased complexity, and we're trying to see if at some point some emergent properties come up on any analytical method that can tell us that this biochemical complex definitely came from a living system versus non-living. And the reason it becomes useful is at some point, we're going to get samples from places where there might or might not be life. It's not going to be terrestrial life. If we find bacteria on Mars, then we know we contaminated Mars, which we probably did anyway. But we're going to bring organics from places like Enceladus or Europa. And we will look for organics. We'll find organics because we know from the spectrophotometry that there are organics in those places. But we don't know if they came from a biosphere or not. And so that's why we need a quantitative definition of life. So I hope I managed to show you some of the coolest applications of this technology. Now, just at the very end, I would like to show you this graph. It shows that the more money we pump into biological R&D, the less drugs we get out of it. So it's basically a reverse of the Moore's law. So something's wrong, right? We're pumping in so much money into biotech and we're getting less and less and less drugs out of it. And so even though that's not really what my lab is working on, that is something that synthetic cell engineers are working on. Just recently, it was demonstrated that synthetic cells can actually shrink a tumor in a living mouse. So it's possible to use synthetic cells as a therapy, and maybe that's possible. It's possible to think of this technology as this new kind of way to address at least some of the problems with the current discrepancy between the amount of money we invest in biotech and the amount of kind of effort we get out of it. So I'm not supposed to have a favorite project, but I'm going to show you my favorite project. This is the first time when synthetic cells, or more specifically cell-free protein expression, was demonstrated under spaceflight conditions. So I was privileged to be part of this amazing team that set out to validate cell-free technologies under spaceflight conditions. So we picked two different, very simple model reactions, a protein expression and a functional RNA transcription. We loaded it up on a sounding rocket. And the reason I'm showing this video is, for one, this is the first time my payload actually went into space. And also I had to be up at 3 a.m. to watch that video. So now everyone has to watch that video. It went up on a rocket. More importantly, it came back. Putting stuff on a rocket is easy. Making the rocket actually not blow up and come back in one piece is the hardest part. So what we did is we recovered the sample, we demonstrated that it does work. The cell-free systems do work under spaceflight conditions. Next step is we're preparing it right now to send and have actual human astronauts perform those experiments on the ISS, and to kind of validate that not only a synthetic cell platform works, but also a synthetic cell platform can communicate with live cell bioreactors to produce useful metabolites from those scalable but less controllable bioreactors. So that would be the end of the science part of my talk. At the end, I would like to show you this, again, a little bit of a provocative slide, but a question. why other fields manage to command large resources for international communities, and synthetic biology is not doing that yet. There are, astronomy, high energy particle physics, they manage to convince their governments that they have this fundamental burning research question, and they manage to build those ginormous facilities. If synthetic cell community and more generally synthetic biology community came together, what would we ask for? I don't have an answer for that. I don't know what would we ask for, but what I want to say is we should ask for something. We should unite our community of all the people that are working on engineering life, engineering living cells. And that's one of the things we're doing is I have the privilege to be one of the co-founders of Build a Cell, which is an international synthetic cell organization of people that believe in being able to build cells from scratch. We're an open community. We're always welcome to have people join. Even if you don't work on engineering cells, if you have any idea, any tool that you think might help with engineering cells, we're always happy to listen. We're always happy to talk to people from other fields. And that would be it. I want to thank everyone from my lab. I want to thank everyone who funded my lab. And I want to thank all the people that in the last almost 4 years of existence of my lab joined. And I want to thank you for listening, and I will take questions. Thank you very much.
Oh, that was a really mind-blowing talk. Thank you so much, Kate. Well, I'm happy to help with asking the questions. And if there's no questions for now, Kate, I would like to ask you, if you had like no risk, unlimited money, and you could have do whatever you wanted, what will be the project you would have start working on?
With no money limitations, I think I would really like to be able to make a universal synthetic cell chassis that can be plugged into any biological system you want. So basically make a synthetic cell that will work under any conditions, within limit, any physical chemical conditions, and make it actually self-replicate and evolve. Right now, synthetic cells don't self-replicate. It's a bug and a feature. For all those biomedical applications, it's definitely a feature because they don't self-replicate. They can't go on and give a patient another cancer like CAR T cells do. But for a lot of applications in studying synthetic biology, studying evolution, we would love the synthetic cells to be able to self-replicate. So that's my wish list, a generic synthetic cell chassis that self-replicates.
Yeah, that would be really amazing. Jan, would you like to unmute yourself and ask the question?
That never works when I teach undergrads. I always say unmute yourself and ask a question, but they still like to type it into the chat.
All right, so I can read it. Thank you for this very interesting talk. Do you think it will be possible to transplant populations of synthetic cells into living organisms to replace hormones or fight cancer? Will they be able to obtain enough nutrition and not cause immune reaction?
The short answer is yes. People are working on it. So the thing about synthetic cells is you make the membrane. And so because you make the membrane, you can decide what you put on the surface of that membrane. And if you make the membrane from either already bioidentical lipids, or if you pegylate the surface of the membrane, you will be able to have synthetic cells that are basically stealth to the immune system. And that's been demonstrated on rodents so far, not on humans, but we have a lot of liposomal drug delivery field to draw on to learn how to make liposomes that are stealth to the immune system. So that's definitely a possibility. The next part will obviously be harder. How do you make synthetic cells that can do something useful? So obviously we're looking at two things, enzyme replacement therapies and cancer. And these are the two areas that people in the synthetic cell community are looking at. It's already been demonstrated that synthetic cells can produce a specific biological that shrinks a tumor in a live mouse without killing the mouse itself. And so it's definitely a possibility. My guess is within a decade or so, we'll start seeing preclinical trials and applications for this technology.
I can see Tomek Włodarski raised his hand. Tomek, do you want to unmute yourself and ask your question?
Okay. Hi. Great talk. My question is, maybe I didn't got this from the beginning about how easy you can modify the composition of the cell. In a sense, can you, for example, study molecular crowding, or how much you can change the ionic concentration inside diffusion of the molecules inside of the cell?
Relatively easy. It depends on the applications, obviously. So one of the things we do in my lab is we modify the cytoplasm. So we modify the sulfate protein expression system. You don't have too much leeway with the ionic strength, because the sulfate expression system has a pretty narrow boundaries under which it functions, but you can modify the external environment. So obviously, because it's a phospholipid bilayer, it's pretty resistant to up to 2-3 moles of salts or ionic gradient across the membrane. And so you can have synthetic cells under a lot of different conditions, and depending on the flexibility of your metabolism, so your synthetic cell cytoplasm, which is a sulfate protein expression system, you can change conditions there. One of the most drastic modifications we've done so far is we made a minimal artificial codon bias. So we basically replaced all of the tRNAs in a sulfate system with artificially transcribed tRNAs only for certain codons, and then we limited the availability of amino acids. We're using it to kind of mimic the evolution of the genetic system, and also to induce an artificial codon bias. This has also been done by other people for other applications, including unnatural amino acids. So there's a lot of work on in the bioengineering community on doing unnatural amino acid expression, and that works much better in synthetic cells because they don't care about endogenous amino acids if you take them away. I don't know if that answered your question fully, or were you asking for more kind of broad set of conditions? Your mouth is moving, but I don't hear anything. I think you're muted.
Sorry, I asked you to unmute. Oh, Gosa, you also, if someone wants to, you have to ask you to unmute.
Yeah, I was not able to unmute myself again. Yes, that's my second question was about the molecular crowding. Can you kind of put a lot of things inside the cell and to study how things move inside, how for example, protein folds inside, yes, on the ribosome. If you put too many things, then maybe the crowding causes the folding to be slower or faster or something like that.
Absolutely. People do that. My lab doesn't really work in this area, but some other people in sulfate and synthetic cell community did a lot of work on molecular crowding and influence of molecular crowding agents on protein expression. You're right that this is a system where you can actually study it. You can induce artificial molecular crowding using either crowding agents or other proteins. And there seemed to be a really narrow range under which most ribosomes are happy. So not enough crowding, your protein expression rates go down. Too much crowding, your protein expression rates go down. And I'm not an expert in this field, so I can't tell you what's the current going theory of why ribosomes are so sensitive to crowding, but they seem to really be.
Aga, would you like to unmute yourself and ask your question?
Was the answer ever yes to that question? Did anyone ever actually?
No, Gosia, as I mentioned, you have to unmute someone if you want someone to be unmuted.
But I think Aga is unmuted. Or it's impossible to unmute. Okay, well, doesn't matter.
That's okay, some people don't like talking on Zoom.
The question is, what's the limitation of synthetic cell scalability?
You can scale the system as much as you want, as long as you have unlimited resources. So by resources, I just mean cash. It takes quite a bit of money to make those systems. This is not cheap work. On a small scale for point of care manufacturing, it is cheaper than whole cells because you can assemble the system faster. But if you want to scale it into a large bioreactors, that's difficult. The biggest cell-free protein expression bioreactor ever made was 100 liters by Sutra Biopharma. And I think they mostly did it to show they can. As far as I know, nobody operates those reactors on more than a few liter scale, and even that's quite a bit by our standards.
And we have one more question about if we can try to mimic life evolution somewhere else, for example, on Mars using the synthetic cells.
Thank you so much. That's a very good question, and the answer is we think yes. So specifically because you brought up Mars, I want to mention a project we're doing in my lab, which is mimicking a regolith, Martian regolith. And if you know, Martian regolith is full of this compound called perchlorate. So we all obviously watched a movie where a guy grows potatoes on Mars, but in reality that would not be possible for the simple reason that Martian regolith is full of perchlorate, which is toxic to terrestrial life. And so you cannot farm Mars unless you find a way to get rid of perchlorate from the regolith. But we can mimic those conditions, and we can try to recreate the prebiotic evolution using the building blocks that we think were possible and accessible on Mars at the time. And we can try to kind of mimic Martian primordial evolution. And people are doing that with better or worse success, obviously. But one of the things that we're definitely looking at right now is using synthetic cells, not just to mimic evolution on Mars, but also to mimic, or other planets, but also to mimic every way in which evolution could work. So basically build some kind of a do-over for evolution. So set up a primordial prebiotic-like evolution scheme, and then see what comes out of it. So basically at which level all those checkpoints that terrestrial evolution took, setting the central dogma, picking amino acids, picking nucleobases, all of that can get a do-over. So we could try to build lifelike systems that don't exactly look like terrestrial biochemistry, that look like the modern life that we know about. So I don't know if that answered the question completely, but definitely the answer is yes. You can do it. It's not done yet. I don't want to say this is a complete. This is completely easy. Building evolution models is always difficult. Building evolution models under conditions that are not the ones of our home planet is even harder. But it's definitely something people are doing.
That is super cool. I would have one more question. What, in your opinion, is the biggest discovery of synthetic life field so far?
I think so far, definitely the work of Craig Venter, where they build a completely artificial genome of a living cell, that was kind of the foundation of the field. And then all the progress, I realize that's not a single discovery, but all the progress on cell-free systems. Basically, what we can do with cell-free systems right now is so much more than we could have done even 10 years ago. So I think it's a lot of technology development. It's not, I can't name like one big flashy discovery yet. I have obviously my favorite ones, but most of I think the best known and the most kind of internationally recognized is the JCDI synthetic cell.
We have one more question in the chat. That's from Parry. What about bioinformatics? How can you build genetic circuits for biocomputation in this system?
Actually relatively easy. The reason the system is being used, not just by my lab, but by others as a chassis for bioinformatics, is because there's no endogenous genetic circuits going on. So what you can do is you can build Boolean logic gates, just like we're doing in bacteria, except they're not leaky, because synthetic cell metabolism can be geared entirely towards building those logic gates and using those logic gates. And so the capacity of the system is similar to the capacity of the bacterial logic gate systems, bacterial bioinformatics systems, but they're much more robust. So basically, if you build a gate, the gate will behave like you designed it, rather than being leaky because of either mutations during the life cycle of the bacteria or some problems with the flux of the reagents through your particular pathway being redirected into the endogenous metabolism. So any rules of the regular cellular bioinformatics apply, except in synthetic cells, most of the time those systems work tighter. Basically, you get a better response curves. I don't know if that answered specifically the question. I always happy to talk more.
Well, I don't see more questions nor comments in the chat. So we will probably slowly wrap up unless someone still wants to. Oh, no, there is one more. From Nina Karpińska. Is knowledge about synthetic cell integrated into medical school curricula?
No, and for the simple reason that there is not much to integrate right now. There is no therapy on the market that is based on synthetic cells. So this is where the field is right now. We're developing those therapies, and I bet you within a decade there will be a preclinical or maybe even clinical trials with synthetic cell therapeutics, but they don't exist yet. So right now there is not much to integrate. Hopefully the people that are graduating from med schools right now will be using those therapies by the time they practice independently, but right now the field is too early yet.
Okay, and I see the raised hand by Monica.
Hello, thanks a lot for the talk. So speaking about bioinformatics, I was wondering if that's also of interest to you, using DNA as a data storage, or if that's a bit of a different field.
That's not something that we do in my lab. We focus on the information processing, so we do the logic gate work, but there is a lot of people working on DNA data processing within the synthetic cell community. Because of my Boolean logic gate work, I'm part of this kind of bioinformatics, biological semiconductor community. And when I go to those meetings, I see a lot of people talking about different ways to store DNA, different ways to pack and read DNA, and how oftentimes it gets integrated into synthetic cells. And this is one of those funny areas where there is a crosstalk between different subdisciplines, because people that do DNA storage need to pack their DNA efficiently. And when we engineer synthetic cells, we also need to pack DNA efficiently to build synthetic cell genomes. And so we're both of those subcommunities are looking at artificial histones, engineering DNA packing solutions, controllable DNA packing solutions. But none of that work is going on in my lab at the time.
Oh, thanks a lot, very interesting.
Thank you.
Okay, last chance for everyone to ask a question to Kate. Well, if there's no more questions, I would like to thank everyone for their attention, and especially Kate for this really amazing talk, opening our perspective for, I guess, future of biology, I would say.
Thanks so much.